So this is where we left off last time. So when it comes to the relationship between the genotype and phenotype, as I've said before, it's not as simple as what Mendel envisioned. It's much more complicated, and we're going to see some of those complications today. So we do know that in general, many characteristics require two alleles, one that came from mom, one that came from dad, but there are other characteristics that involve multiple genes and multiple alleles. And sometimes one gene can affect the expression of another gene. So, but even though the phenotype may not be what is expected, segregation and independent assortment are still happening. It's just that we don't see the result in the phenotype. So we have cases where alleles may not be completely dominant or recessive. Genes have more than two alleles, so there are some genes that have multiple alleles. And when one gene produces multiple phenotypes, so a single gene can affect several observable characteristics. We're going to see examples of these. So complete dominance is basically what we have seen in our examples of the peas, where we have one phenotype completely dominant over the other. So like the purple flowers were dominant to the white flowers. So that even a heterozygote, like big P, little p, is still purple, right? Because the purple allele was dominant to the white, and the white allele was recessive. There are cases of incomplete dominance where the phenotype in the hybrids is intermediate. So it doesn't look like either one of the parents, but it's an intermediate phenotype. But when we talk about incomplete dominance, we are talking about the phenotype being incompletely dominant, not the actual expression of the alleles. It's just what we see is incompletely dominant. And in the case of codominance, both alleles are expressed and we see both characteristics. So neither allele is dominant or recessive, and we see the two phenotypes. They're both expressed. So here we have some flowers, red flowers and white flowers. When we cross our two parental strains, and remember these are true breeding strains, so red mated with red produces red, white mated with white produces white flowers. But when we cross the red and the white flowers together, we get pink. So the offspring are pink. So they're neither red nor white. It is an intermediate phenotype. So we call this incomplete dominance. But it's just the phenotype. It's not the alleles. They're both being expressed equally. It's just that what we see, what we perceive, is an intermediate phenotype. So it's like a blending, in this case, of the red pigments and the white pigments. So the red pigment is diluted, and so it appears pink. But both alleles are being expressed at the molecular level. It's just that the observed phenotype is intermediate. So we call this incomplete dominance. Notice how they tend to write this, where in this case the gene for the flower color is denoted by a capital C, and then they put, exponents on them to indicate what alleles are there. And so notice that in both cases, the alleles are written in capital letters, right? Because neither allele is recessive. So some people might argue, well, then isn't this an example of codominance? Because we write the codominant alleles the same way. And no, because by definition, incomplete dominance is referring to the phenotype, what we see. At the molecular level, both genes are equally expressed. So in that sense, both alleles are codominant, but the phenotype is intermediate, so we call that incomplete dominance. When it comes to true codominance, we see both phenotypes, right? They're both expressed. So here we see neither the red phenotype nor the white. We see pink, just intermediate. So they call this incomplete dominance. But incomplete dominance just refers to the phenotype, what you can see. And if we cross two of the F1s, right, two of the pink flowers, we get about 25% red, 50% pink, 25% white. So you can see how that Punnett square was done. Remember that the alleles are written along with the gene, which they're using C to indicate the gene for flower color. And then they put the type of allele on of the big C. So RR will give you the red flower. RW gives you the pink flower, and WW gives you the white. But in this case, white is not recessive. White is expressed equally, right? Just as the red will be expressed. But if you have a red allele and a white allele, you see an intermediate phenotype. So like I said, it's a blending of the red and white pigments to give you pink. So both pigments are there, but we perceive them as pink, as an intermediate phenotype. So incomplete dominance just refers to the phenotype, not the genotype. So the relation between dominance and phenotype. So a dominant allele, it's not subduing or, it's not impacting, the recessive allele. The recessive allele is still being expressed. It's just that you don't see it if you have a dominant allele. Alleles are just variations in a gene's nucleotide sequence. So it's still the gene. So it's still the gene for flower color, but you have an allele that makes red flowers, an allele that makes white flowers. It's still the gene for flower color. There's just different versions of that gene. And for any character, for any trait, dominance and recessiveness relationships of alleles will depend on the level at which we examine the phenotype. So what do we mean by that? So this disease, Tay-Sachs disease, is a fatal disease, unfortunately. A dysfunctional enzyme causes an accumulation of lipids, right, of fats in the brain. Now the brain already does have a lot of fat in it, right, because all the neurons are wrapped with the myelin sheaths, which are, lipids. So there's already a lot of fat in the brain, but excess fat leads to the death of the brain. So if we look at the organismal level, the allele is recessive. So that means you have to have two recessive copies of the Tay-Sachs allele in order to express this trait. If you're heterozygous, one normal allele and one Tay-Sachs allele, you're normal, right? That normal allele produces enough of whatever enzyme or protein that's needed to remove the excess fat that you're okay. At the biochemical level, the phenotype is incompletely dominant because you have the good allele producing the good enzyme, and the bad allele is producing the bad enzyme. So both enzymes are being produced equally. It's just the bad enzyme doesn't work. At the molecular level, the alleles are codominant because they're both being expressed. They're both being transcribed and translated to make their respective enzymes. It's just that only one of the enzymes is good and the other one is bad. So whether you talk about a characteristic as being dominant, incompletely dominant, or codominant, it may be beneficial to make it clear at what level you're discussing that dominance. Because in general, if you talk about an incomplete dominance situation, you're talking about the phenotype being intermediate. What we see is intermediate. If you have codominance and you see both phenotypes, that's how we typically define a codominant trait. But as we've seen here, if you're discussing at the organismal level, the biochemical level, or the molecular level, the dominance is different. So it does get sort of tricky, and that's why in science we try to be precise and say exactly what we mean. And so you should always try to avoid using the word it, because it is non-specific, right? I don't know what it is, right, that you are referring to. So let's always try to be precise and say exactly what we mean. You don't leave it up to my interpretation, because my interpretation of your answer may be different than your interpretation. All right, so always try to be precise. But when it comes to dominance, again, in general, incomplete dominance refers to a phenotype that's intermediate between the two parents. Codominance also refers to the phenotype because we see both phenotypes. They're both expressed, and we see them both. But alleles at the molecular level, they're always being transcribed and translated. So in the case of Tay-Sachs, your good allele is making the enzyme you need to remove that fat from the brain, and the bad allele is making an enzyme that doesn't do anything. Right? So they're both being expressed equally. At the molecular level, they're both codominant because they're both being expressed equally. So when it comes to dominance, you do have to try and qualify exactly what you mean. But the traditional definitions are what we saw before, with the pink flowers being an incomplete phenotype. And we haven't seen a good example of codominance yet, but when we do the genetics problems later on, you'll have some examples of those. So dominant alleles are not necessarily more common in populations than recessive alleles, although in general they are. The dominant trait is the one you see the most. But extra fingers and toes is actually a dominant characteristic. It's just that very few people have it. Most people are recessive, for 5 fingers and toes on each hand and foot. But this does happen. When it comes to the feet, the extra toes, those are typically surgically removed, during infancy, because it's hard to get shoes, right, that will fit, and they have to be custom made, and that leads to all kinds of problems if you don't have a good fitting shoe. So typically they tend to surgically remove the extra toes. But with extra fingers, some people are, just choose to keep them, because if you got 6 fingers on each hand, it's like, so what? It doesn't affect how you can do things with your hands. So when it comes to the fingers, as long as they're all functional, most people with extra fingers will keep them. Extra toes, they like to get removed because it's just too hard on the feet to wear shoes and things with extra toes.
Is that picture of 8 toes on each foot real?
Can you say that again?
Is that picture of 8 toes on each foot real?
Yes.
Oh my God.
Yes, it's real. So, but a lot of those toes are going to be removed because it's just not, you don't need extra toes. You're not going to use those for anything. And like I said, it's just much harder when it comes to getting shoes and things when you have extra toes. And so those will be removed. But this guy here, he's not going to have an extra finger removed because they all work just fine. So there's no need. So again, in this case, extra fingers and toes is a dominant characteristic, but the majority of the population is recessive, for 5 fingers and 5 toes. So most traits, that are the most common in populations are dominant characteristics, but not always. A rare example. I don't think I've ever seen anyone with extra fingers or toes, but I mean, they're out there. When it comes to multiple alleles, most genes exist in populations in more than 2 allelic forms. So there's more than just 2 alleles. A good example, blood type. So when it comes to the ABO blood group, the A and B antigens are extremely important when it comes to transfusing blood. So if you have the gene for the A antigen, you have type A blood. If you have the gene for the B antigen, you're type B. If you have both genes for A and B, you're type AB. And if you have neither gene, you're type O. So O blood does not have the A or the B antigen. And antigens are substances that the immune system sees as foreign and reacts to them. Most commonly, antigens are proteins, but they can also be carbohydrates. In the case of the AB antigen, it is a carbohydrate. So when they type blood for transfusion, they have to type for the A and B antigens, as well as the Rh antigen, which I'll talk about in a minute. Those are the most important when it comes to the immune system, because if you do not have these antigens on your red blood cells and they transfuse you with blood that has these antigens, your immune system will attack and destroy that blood and you'll die. Usually not right away. You can live for a week or more before you get sick and die because the immune system takes a little time to respond. So imagine what happened when humans first started to transfuse blood. Sometimes it worked, sometimes it didn't. And then they had to start looking for why. And they discovered these antigens. And in particular, the A and B and the Rh antigen are extremely important when it comes to blood transfusions. So if you have type A blood, you can either be homozygous, AA, or you're heterozygous. One big A and then the little I is what they indicate for neither A nor B. If you're type B, you can be homozygous and have 2 alleles for the B antigen, or only have 1 allele, and you still produce that antigen. If you have the alleles for both A and B, you make both antigens. If you have neither the A and B allele, then you don't produce any antigen, so they call that type O. Now the Rh antigen, and Rh stands for rhesus, and not Reese's peanut butter cups, but rhesus as in rhesus monkey. So it's called the rhesus factor. And if you have it, you're Rh positive. If you don't, you're Rh negative. But again, the Rh antigen is something that the immune system will aggressively attack if you do not possess the Rh antigen. So when it comes to to blood donation, who is considered the universal donor?
O negative.
Yeah, not just O, but O negative, because O negative blood has none of the offending antigens, right? No A, no B, no Rh antigen. So O negative is the universal donor. And so they always ask for people with type O blood to donate, because O negative is the best. The second best is O positive, right, which is what my blood type is, O positive. But they always want type O donors because type O can be given to almost everyone, especially if it's O negative. And then who is the universal recipient?
AB positive.
Right, AB positive, because they have all the antigens. So they can receive any blood. A blood, B blood, O blood, positive, negative, all of it. But AB positive does tend to be the least common blood type in most populations, whereas type O, there's usually a very large percentage of the population that's type O in almost everywhere. So they always want type O blood donations. And if you're O negative, you're the best. Everyone can take your blood. O positive is second best. So do we all know our blood type? If not, why not? Why don't you know your blood type? Ask your doctor. Ask them to type your blood if they don't have it on file, right? So you know what it is. Or go donate blood. If you donate blood, they will type it for you and they'll give you a card which has your blood type on it. So it's good to know what your blood type is, right? Because maybe someone needs an emergency transfusion. You're the only one available, right? So you could donate. Yeah, so you should know what your blood type is. You know, it's a fun thing, right? And it's also important for the ladies, if you're Rh negative and daddy is Rh positive and your fetus is Rh positive, that can lead to complications causing miscarriages. So it is good to know what your blood type is and what your partner's blood type is. But this is an example of multiple alleles. And the A and B antigens are co-dominant, right? Because if you have the A allele and the B allele, you express both of them. You find both antigens on your red blood cells, so they're co-dominant. Now, pleiotropy is a case where a gene has multiple phenotypic effects. So you see multiple phenotypes as the result of a single gene. An example is albinism. So I know you've all heard of albinos, right? So albinism involves one mutated gene for one enzyme. And it's because melanin production, right? Melanin is the brown pigment, right? So everyone except albinos produce some melanin. Some people produce a lot of melanin and have dark skin. Others don't produce very much and have light skin until we get sun exposure, and then we tan, so we start producing more melanin to protect us from the UV. But if you cannot produce any melanin, you're an albino. And that's caused by one mutated gene and one defective enzyme. Now, you do have to be homozygous, right? So you have to have two defective alleles, so you don't produce any melanin. But because there's no melanin produced at all, you see the phenotype in the hair, in the eyes, in the skin. So 3 phenotypes, 1 gene. So it's pleiotropy. So I have some pictures. This technically is not an albino peacock. It's just a white peacock, but it looks cool because it's all white. But I don't think it's technically albino, but it is pretty. Here we have some albino animals, an albino squirrel, a deer, a cobra, and an alligator, or a crocodile. I think it's an alligator. So these do happen in nature, right? So you have these mutations that occur, you know, often just randomly, and you end up with an albino. How often do you see albino animals in nature, and why not? In DC, really? But it's pretty rare to see albino animals in nature. Why is it rare? why don't we have populations of albino squirrels and albino deer? And right, no camouflage. So most animals, their coat color, their skin color is designed to help them to blend into the background, or at least not to stand out. But if you're albino, it's like having a bullseye, painted on you. You're very easy to see by a predator. And so albino animals typically do not live long enough, at least not long enough to reproduce. So they're often kept in zoos, when they're discovered, so they can be kept alive and people can see them. But albinism occurs in all different kinds of animals, including humans. So all groups of humans, black, white, Asian, Hispanic, you can find albinos in every population of humans. It's most noticeable in Africans and people of African descent because they have no brown pigment, they have no melanin. But you find white people that are albinos, Asians that are albinos, and of course, Africans that are albinos. Right? They just simply cannot produce any melanin. And so we see multiple phenotypes in the hair, skin, and eyes. Now, these people do need to take extreme precautions when being outdoors. They need to wear sunglasses. They need to wear hats, right? They need to cover their skin or use lots of sunscreen because they don't have any melanin to help protect them from the UV. And so they are at higher risk of a premature aging, skin cancer, and cataracts. So they do need to take extra precautions if they're going to be outside. Now, eye color. Unfortunately, these diagrams that I've been using are actually outdated. But the point here is that eye color, mostly controlled by 2 genes, but there may be up to 14 genes that can influence eye color. You'll see some combinations here, but this is outdated because you notice here at the bottom, we got 2 blue eyes, and it says a 0% chance of brown eyes, and that's not correct. I don't know what the exact percentage is, but 2 blue-eyed parents can produce brown-eyed children, and we'll see that in a couple slides. But eye color has multiple genes and multiple alleles. And so you can get all different kinds of variations, brown, green, blue, hazel eyes. But even weirder things can happen. Is anybody here a baseball fan? You watch baseball? In particular, the Nationals? Nobody cares about baseball? Well, they have a pitcher, Max Scherzer. Look at his eyes. So he has a pretty rare condition called heterochromia. So 2 different colors. So his eyes are 2 different colors. And this is most often a genetic characteristic. I don't know whether he's passed it on to any of his children, but it's a very interesting, look, right, to stare. And of course, he's got his eyes wide, so you can really see the difference in the eyes here. So weird things can happen, and they happen in other animals as well, right? So we got a cat with 2 different color eyes. We have dogs with 2 different color eyes too, right? So we do see this in different kinds of animals. Now, and this is just the FYI thing, because for a long time they did not know this. And I forget exactly when they discovered this. It hasn't been all that long, maybe within the past 10 years or so. But on chromosome 15, there are 2 important genes for eye color, the OCA2 gene and the HERK2 gene. And so the protein produced from the OCA2 gene is known as the P protein. It's involved in the maturation of melanosomes. And melanosomes are organelles that contain melanin, right? They store the melanin. And then the melanin can be deposited, in the skin or the eyes or the hair. In this case, we're talking about the eyes. And so the P protein plays a crucial role in the amount and quality of the melanin that's present in the iris. Less P protein means less melanin, so you get blue eyes instead of brown. The HERK2 gene, it controls the activity or the expression of the OCA2 gene. So turning it on and off as needed. So these two genes are very important for eye color, right? In particular for having brown eyes. So if you have one of these two genes that is defective, you have blue eyes, right? So if you have a defective OCA2 gene, you have blue eyes. A defective HERK2 gene, you have blue eyes. So if you have two parents that have blue eyes, but each parent has a different defective gene, their children can each get a good gene. And so the children can have brown eyes because they inherited the good gene from one parent and the other good gene from the other parent. So the child has both good genes, but the parents only have one good gene, but they have the opposite one. And so it is possible for blue-eyed parents to have brown-eyed children, but this is something that they did not know for a long time. They just assumed blue is recessive and brown is dominant. So it was not possible for blue-eyed parents to have a brown-eyed child because they should be homozygous recessive. And so you only get recessive offspring. But they did not understand all the genes that were involved in eye color. Now they do. This may have caused some problems in some marriages where you had two blue-eyed parents and had a brown-eyed child, and they thought the only way for that to happen is if mom had cheated on dad and got pregnant from a brown-eyed guy. So we're learning new things all the time about genes and genetics. And this is a fairly recent discovery. Some traits are determined by two or more genes. And of course, those two or more genes have two or more alleles. Now, epistasis is when a gene at one locus, so at one location on a chromosome, alters the phenotypic expression of a gene at a second location or second locus. So one gene is doing something to affect the expression of the phenotype that would be produced by that second gene. And so you may not see the actual phenotype because one gene is having an epistatic effect on another gene. So albinism was one example because even though a person has the genes that should give them brown skin or brown eyes or brown hair, that defective enzyme prevents brown skin, brown eyes, and brown hair. When it comes to baldness, if you have the gene for baldness, your hair falls out. But you have genes for hair and genes for hair color, but the baldness gene is neutralizing that phenotype. You can no longer see it because your hair has fallen out. So when it comes to certain animals like mice and other mammals, coat color depends on two genes. One gene will determine the actual pigment color. And so we're going to use a big B for black and little b for brown. And the other gene with alleles C for color, big C for color and little c for no color, that determines whether the pigment will be deposited in the hair. So in this case, for many animals, their hair color, their coat color, they have one gene which is producing the melanin, another gene which determines if the melanin gets deposited into the hair shaft, right, to give the hair the dark color. So we're going to see that an animal may have the gene, right, for producing melanin, but the other gene to put the melanin into the hair is defective. So here we have some mice. Remember, big B codes for lots of melanin, right? So in theory, if you have a big B allele, you should be black, right? You should have a black coat color. But remember, the gene, the C gene, is needed to actually deposit the melanin into the hair shaft so you can see that color. So if you have two little b's, you produce less melanin, so it's a brown color instead of the dark brown black color. And really, black is usually just very dark brown. So it's not actually black, it's just really dark brown, but it looks black to us. So a big B and a big C means the animal has a black coat. So as long as you have one big B and one big C, a black coat color. If you have two little b's, right, the recessive condition, and a big C, you'll have a brown coat color because you are still producing, some melanin. It is being deposited into the hair shaft, and so you can see the color. But you have to have at least one big C allele in order to have any color in the coat. So in this case here, all these white mice, you got two big B's, right? They're making lots of melanin. It's just that they've got two little C alleles, so they cannot actually deposit the melanin into the hair shaft, right? So the hairs are not pigmented. Even though they got the alleles, right, to make all that melanin, it can't be deposited into the hair. So here we got two little b's, which should be brown, but no big C alleles. So again, no pigment can be deposited into the hair. So again, this is not something that Mendel could ever have imagined would happen, but we have cases where the expression of one gene affects the phenotypic expression of another gene. So any questions on this? Right, so remember, epistasis, one gene is affecting the phenotypic expression of another gene. So in the case of the coat color here, you must have the gene to deposit the melanin, to deposit the pigment into the hair shaft, right? So the hair has color. If you don't have that, it doesn't matter how much melanin you're producing, it's not going to be deposited into the hair, and so you won't see it. So epistasis, one gene affects the phenotypic expression of another gene. That other gene is still being expressed at the molecular level. It's making its protein, enzyme, whatever it needs to do. It's just that you can't see it because another gene is affecting that expression, right? What we see. No, they don't necessarily need to be near each other, even on the same chromosome. Now, quantitative characters are those that vary along a continuum. So often we see this with polygenic inheritance, where you have two or more genes affecting a single phenotype. So the most common example is skin color. So multiple genes, multiple alleles, and so you get every possible combination. So very, very Caucasian, right? Light-skinned, and then a very dark brown skin. And so every possible variation in between, because there are multiple genes and multiple alleles. So you can get children that are either lighter or darker than either parent, right? Because of whatever combination of genes and alleles randomly occur in that child. And so we see all different skin tones, And that's because it's a polygenic trait. Multiple genes, multiple alleles. So you get a many different degrees, right? So a scale, right, from very light to very dark. And it's just melanin, right? It's just melanin. Some people make a lot of melanin and some people don't, but it's just pigment. Now, another departure from Mendelian genetics, so again, something that Mendel could not predict from his peas, is how the environment can affect, right, the phenotype. So the norm of reaction is the phenotypic range of a genotype influenced by the environment. An example, hydrangea flowers, which most of you probably don't know what hydrangeas look like, but it's an old-fashioned kind of flower. And the old-fashioned kind had either blue flowers or pink flowers or something in between, depending on the soil acidity. So in this case, the pH of the soil would determine if your hydrangea had blue flowers or pink flowers or a really beautiful mix of, like, blue, purple, pink on the same bush. It's really very pretty when you see something like that. But it's the exact same plant, exact same genotype. It's just the soil pH determines whether the flowers are red or blue. Or if the pH is around neutral, you get these purples. So really beautiful. Now, when it comes to the soil pH, it's the opposite of pH paper. So if you're familiar with pH paper, like what color does pH paper turn for acid? Red. And for base, it's blue. It's the opposite for the hydrangeas. So the red pink flowers are alkaline soil, above pH 7, and the blue flowers are acidic soil, below pH 7. So, but it's interesting because people, will sometimes decide to change the color of their hydrangeas by changing the soil pH around them. So maybe one year they want pink, another year they want blue, or something in between. So you can change your soil pH, and that will affect the colors. I have a bunch of hydrangeas on my property. Most of them are blue. I got one that's pink, and I have one that is sort of the purplish one, so it's got a more neutral pH. So it's very interesting. And I've done nothing to them. It's just whatever the soil pH is where they're growing. So norms of reaction are generally at the broadest for polygenic characters. So again, multiple genes and multiple alleles. And such characters or traits are called multifactorial because both genes and the environment affect the phenotype. So you may have heard the expression nature versus nurture. So nature means your genetics, nurture means the environment. So our environment can influence our genetics, right? 'Cause we can't change our genetics, but we can influence them. So for example, let's say that you have a family history of heart disease. Well, you can influence those genes by eating a healthy diet, right? Low in saturated fat, low in cholesterol, and by exercising. Of course, you have to want to do that. But you can modify, right, your genetics by influencing it, change the environments, right? Because you can't really change your genetics, but if you change the environment, that can improve a potentially negative genetic outcome. An organism's phenotype includes its physical appearance, internal anatomy, its physiology, and behavior. So a behavior is a phenotype. and behaviors are determined genetically, but they can be influenced by the environment. So an organism's phenotype will reflect its overall genotype and its unique environmental history. So we're not all exposed to the exact same things, the exact same environment, but that can affect how we behave, right? It can affect various other phenotypes. Humans are not good subjects for genetic research, right? One, our generation time is too long. It takes too long to create a baby. It takes too long for that baby to reach sexual maturity and then have more offspring, right? Have more babies. Parents produce relatively few offspring, right? Most of the time you have one baby at a time, unless you take fertility drugs, but still, most humans do not have very many children. And of course, we cannot selectively breed humans to see what would happen. Although it's been tried, before, but we can't use humans for genetic experiments. It's illegal and unethical. But we can use plants and animals, right, and see what happens to them. So Mendelian genetics is the basis for all genetics, but as I keep saying, it's way more complicated than Mendel could ever have imagined, right? Because he didn't know about DNA or genes or chromosomes, epistasis, polygenic inheritance, any of that kind of stuff. He just experimented with peat plants. So he learned a lot. He learned the basics. And so he is considered by many to be the father of modern genetics. Even though he knew almost nothing about actual genetics, he did learn a lot. And he made a lot of observations. He recognized patterns, right? Our 3-to-1 phenotypic ratios. So he did a lot to start the field of genetics. And he was a monk, right? A religious guy, not a scientist. So we do like to look at the genetics of humans, especially if we're trying to anticipate possible genetic problems that might occur. So we use pedigree analysis. A pedigree is a family tree, which shows the interrelationships between the parents and the children and the various genetic conditions or traits that they may possess. And so we can use those inheritance patterns to help determine, probability of what will happen in the offspring. So when we write a pedigree, we have some standard symbols we use. So males are squares and females are circles. Unaffected males and females just have a white square or circle, and the affected ones are shaded. We use lines between males and females to indicate that mating, right, has occurred. And then the offspring are listed in the order in which they are born. Firstborn left, lastborn on the right. So here we're looking at a genetic trait called a widow's peak. And so if your hair comes down to a point on the forehead, that is a widow's peak. Some people have it, some people don't. Sort of like, you guys are way too young for this, but the classic example was the old TV shows, The Munsters, with Eddie Munster, which had a very pronounced widow's peak enhanced by makeup. So you can look at the pedigree, and the people who have the widow's peak, they're shaded, and the ones without it are white or clear. And then that can help you to determine if the trait is dominant or recessive. And so when it comes to a widow's peak, is it a dominant or a recessive trait? It's dominant, right? So it's a dominant characteristic. If you have the allele for the widow's peak, then you'll have it. If you're homozygous recessive, then you do not have it. Now, this is a trait that is also affected by epistasis. If you have the gene for baldness, even if you had a widow's peak, you can't see it, right? Because the hair fell out. So another example of epistasis. So you have the gene to produce a widow's peak, but the gene for baldness prevents you from seeing that phenotype. So that's epistasis. When it comes to earlobes, some people have attached earlobes. So the earlobe is stuck to the side of the head. Others have a detached earlobe. There's actually a gene which some people have that tells the cells along the side of the head to self-destruct, and that allows the earlobe to detach. I have attached earlobes, but looking at this pedigree, are detached earlobes dominant or recessive? It's recessive, so both parents have to carry the recessive allele. They do not have to have attached earlobes. They could be heterozygous. And so they have a normal, or in this case, a free earlobe allele, and they have an allele for an attached earlobe. But you have to have both of the recessive alleles to have an attached earlobe. If you have just one of the dominant alleles, your earlobes are free. And so in class, it's really fun to look at everyone around the room and see what kinds of characteristics they have, like a widow's peak, or an earlobe, or a tongue rolling your tongue. Do you know what I mean when I say rolling your tongue? If you can roll your tongue, you can do this. Yes, I'm sticking my tongue out at you. You can curl it up on both sides. Can anybody roll their tongue?
Me.
Yeah. Right. So some people can do it and some people can't. Right. So it depends on whether or not you have that particular allele. Right. So some can and some cannot. The ability to taste bitter. That's really interesting. Some people have really good tongue control, right? But the ability to taste bitter is not something that everyone has. So you don't realize that not everyone can taste bitter. And so there is a type of paper with a chemical on it called PTC paper, and there's others, but PTC was one that was used for a long time. And so we would have students take a little piece of this, and then they would taste it. And you could tell by the look on their faces whether or not they could taste it, because it's nasty and bitter tasting. But then there were some students who are like, I just taste paper, right? They don't taste anything. All right, so not everyone can taste the same. And so some people like me hate things like beer. Beer is bitter. It is just bitter and nasty. But a lot of people cannot taste the bitter. And so beer tastes good to them. And they can't understand why some people, can't stand the taste of alcohol, because it's bitter. And if you're a bitter taster, alcohol doesn't taste good. Beer doesn't taste good. It tastes awful to me because it's so bitter. But lots of people, drink it like, water or soda, and it's like it's awful. It just tastes awful, but they can't taste it. And they don't realize that not everyone tastes the same. Or when it comes to a sense of smell, hydrogen sulfide, the rotten egg smell, the sulfur smell, some people can't smell it. Other people can, but some people can't. And so those people that can't smell it, unfortunately, cannot smell their own body odor. And so they don't realize that they stink because they can't smell it. So it's really weird, but not everyone can smell sulfur compounds. And so, they don't realize that they stink. Everyone else does, and no one wants to say anything, but they stink, and they just can't smell sulfur. So these are all different traits, right, caused by different genes and different alleles, but not everyone has the same ability to taste, the same ability to smell things, right? So there really are a lot of variations among the human population. When it comes to genetic disorders, they do tend to be recessive, which means you have to have both of the recessive alleles to express the disorder. Like we talked about with Tay-Sachs disease, you have to have 2 recessive alleles to have Tay-Sachs disease, and then that's very bad because it's fatal. 1 good allele, 1 bad allele, and you're okay. But you're a carrier, right? So you carry that bad allele. If you mate with someone who's also a carrier, then there's a chance that your child can have that disease condition and die. So you only see the recessive characteristic, that genetic disorder, if you're homozygous recessive. If you're heterozygous, you're phenotypically normal, but you are a carrier. So you have the chance of passing on that trait to your offspring. Albinism is a recessive condition, so you have to have 2 recessive alleles to be an albino. 1 good allele, 1 bad allele, and you have relatively normal pigment, so there's no one would notice anything unusual about you. So here we see, and we're just using A's to indicate the gene. So the big A is the normal allele, and the little a is the recessive allele. If you have 2 heterozygous parents, they have normal pigment. And what we notice when we do the Punnett square, we have a 3-to-1 ratio, because remember, 2 heterozygous parents, this is a monohybrid cross. And so we've learned there's a 3-to-1 phenotypic ratio. So 75% of the offspring will have normal pigment. 25% will be albino, right? Because there's a 1 in 4 chance of getting those 2 recessive alleles. Now the genotypic ratio is 1 to 2 to 1, right? Because that's the characteristic genotypic ratio or frequency for a monohybrid cross. So again, if 2 people are heterozygous, they have a 25% chance. So in general, the odds of having that homozygous recessive child, they do tend to be low. Now, matings between very close relatives, like first cousins or brothers and sisters, are called consanguineous matings. So these increase the chance of 2 carriers coming together and producing that diseased offspring. So most societies and cultures do have taboos against consanguineous matings. So no brother sister, no mother son, no father daughter, right? No first cousins, because that increases the risk of combining those bad genes in the offspring. Animals in nature, after the offspring are weaned, right, when they're old enough to start eating solid foods and hunt or gather food on their own, they are driven away from the parents, right? They're forced to leave. And that reduces the likelihood of these consanguineous matings here. So the offspring, once they're old enough, they're kicked out, right? Like what some parents do with their kids. You're 18, get out, get a job, get on your own, right? But many animals do this in order to prevent, siblings from encountering each other and mating with each other. It still does happen every now and then, right? But it reduces the chances when you force the offspring away. But the reason why humans have laws against these kinds of matings and why they have taboos against them is not because of issues of morality. It's because of genetics. Humans learned a long time ago that very close matings between closely related humans results in bad genes and bad characteristics appearing in the offspring, right? So it's not an issue of morality. It's an issue of genetics. And that's why humans do this, although over the centuries, it's become an issue of morality, but it never was. It was about genetics. Even though they didn't understand genetics, they could see, we've got a sick kid here, right? Because we're closely related. And we've seen these kinds of things in the royal families, because remember those countries that have royal families, you were only allowed to marry other royalty. And since there were very few members of royal families, that resulted in a lot of cousins, mating, right? So we see a lot of genetic diseases. The most common one historically was hemophilia, right? Bleeder's disease, right? Where those individuals do not produce clotting factor. And so when they get injured, it takes a long time for them to stop bleeding. So they often would die from blood loss. And we saw that a lot in royal families because they had a lot of close matings, right? Because you had to marry someone who was royal, and there weren't that many royals. In other religious groups, where they tend to only marry and mate with members of their own particular, tribe, their own particular religious group, we see a lot of these kinds of things. In Ashkenazi Jews, we see very high incidence of Tay-Sachs disease and some other genetic conditions that you don't find in any other populations because they have a small, breeding population. They only mate with members of their own tribe, their own religious group. And that concentrates all these bad genes. So like I've said before, outbreeding, breeding with members, of different races and ethnicities, right? Different tribes, that's what is good, for the species. It increases diversity and variation. And now we see it decreases the chances of bad genes coming together. So it is a good thing, right, to mate with someone who's very different from you because there is far less of a chance of any bad genes coming together. And it also increases variation, which is good for the species. So consider, marrying someone who's not like you, right? Because you'll have a much healthier child, right? Hybrids express what's called hybrid vigor, right? Because they tend to get the best characteristics. And so they tend to be, the best adapted or suited to survive. So it's something to think about, for the future. Now, sickle cell disease is a disease that typically affects Africans or people of African descent. Here in the US, it affects about 1 out of 400 African Americans. And so sickle cell disease occurs where the white blood cells assume a sickle shape, so kind of like a crescent moon shape. And that's an abnormal shape. And those red blood cells get stuck in capillaries and clog the capillaries and prevents normal blood cells from being able to get in there and deliver the oxygen and remove the CO2, and it can lead to organ damage and organ failure and death. It's caused by a single amino acid substitution in the hemoglobin protein. And so as a result, that would change the shape of the red blood cell and affect its ability to carry oxygen. And the symptoms include a physical weakness, pain, organ damage, paralysis, in severe cases, death. With modern medicine, now though, we can help someone who does have sickle cell disease to live a lot longer. I did have a student one time. I'd say she was probably late 40s, around 50 years old, and she had a sickle cell anemia, right? The full sickle cell. And what she would do is drink a lot of water, a lot of fluids, right, to keep her blood volume up, which she believed helped her condition. And we were able, because back then we were still allowed to make blood slides in class. And so I did get the little lancet, to prick her finger so we can make a slide. And so all the students were able to see real sickle cells, for real. So from one of their classmates. So it was pretty interesting to actually see it for real sickle blood cells. But she would just drink a lot of fluid as a way to help deal with it. But it is a potentially very serious and fatal condition. Although today, modern medicine can help if there's a crisis to help that person to survive. But the sickle cell trait evolved for a very specific purpose. It's actually, the trait is beneficial, not the actual disease, but if you're heterozygous, one normal hemoglobin allele and one sickle cell allele, that actually protects you against malaria. And so that's why this evolved in Africa, because you had high incidence of malaria. So evolution said, okay, malaria is very debilitating. It kills a lot of people. A random chance mutation occurred, which caused the sickle cell trait. But if you're heterozygous, one good allele, one sickle cell allele, you're protected from malaria. If you're unfortunate enough to get both sickle cell alleles, then you would die. But that would only happen in about 25% of the population. And so the other 75% would get protected. Now, let's see. Yeah, we'll do the Punnett square. So, but it's estimated that about 4.4 million people have sickle cell disease, so the actual disease. And then 43 million have the sickle cell trait. About 80% of sickle cell disease cases are believed to occur in sub-Saharan Africa, mostly Nigeria, the Democratic Republic of Congo. And in 2015, it did result in over 100,000 deaths. So that is a significant mortality. But as I said, if you're heterozygous, you're protected from malaria. And malaria killed more people, than sickle cell disease does. And so it was a beneficial characteristic. So let's look at a Punnett square. I don't think I have a blank. Let me see if I have a blank slide. No. So let's use big S is the normal hemoglobin, and little s is the sickle, right? So whenever you do a genetics problem, always write out everything you know. And so we know that if you're homozygous, you're normal. If you're heterozygous, you're normal, but a carrier. And if you're homozygous recessive, you have a sickle cell disease. right? So that's what we know. So if you're heterozygous, right, you're going to survive malaria. So let's take two heterozygous individuals and cross them together. And so again, we are going to have a monohybrid cross. So you already know what that means. So one heterozygous individual and the other heterozygous individual. And those are the gametes produced by them. Half the gametes will have the normal allele, and the other half will have the sickle allele. So what we see is that 75% of the population will be phenotypically normal, right? Our 3-to-1 ratio, and 25% for the sickle disease, and they will typically die. But the majority of the population is protected from malaria. And so these 25%, which are susceptible to malaria, they still get protected because malaria is transmitted by mosquitoes. And if the majority of the population are resistant to malaria, you have more people that can be bitten by the mosquitoes, right, and survive and not get malaria. And so there's more, bodies for the mosquitoes to choose from. And so they're less likely to choose you if you are homozygous normal. So you get protected, right, by the rest of these. Oh, so you're heterozygous, Demilula?
Yes, I mean, I'm Nigerian, so all of that like very, very familiar to me.
Yes, but here's an example of evolution in action. Malaria was causing a lot of deaths of humans, so a mutation occurred which protected them against malaria. Yes, there was a negative consequence, but far fewer people would die from sickle cell disease than they would from malaria. So evolution resulted in a mutation that would protect the majority of the human beings living in an area that had a lot of malaria. Now, unfortunately, today, since malaria is more treatable, not as common, human beings can do a lot more to protect themselves. Having the sickle cell trait is actually becoming more of a negative thing, right? Because malaria is not as big of a problem as it was during the evolution of humans. And so because now we have, good ways to deal with mosquitoes and other medical treatments for malaria. So the sickle cell trait is less beneficial today than it used to be, only because malaria is not as bad of a problem as it used to be. It's still a problem, but it's not as bad. So, but evolution finds a way. So some human disorders are caused by dominant alleles. Dominant alleles that cause a lethal disease are rare because if you have that allele, you have that disease and you're going to die. And so you're not going to survive long enough to pass that on to your offspring, usually. So very often these dominant characteristics that are lethal are the result of a mutation. Now, achondroplasia is a form of dwarfism caused by a rare dominant allele. So it's a dominant allele for the most common type of dwarfism. So these are little people. And for achondroplasia, their long bones do not grow. And so that's why they're very short in stature, but their heads are normal size. And most of their internal organs tend to be fairly normal in size, which creates a problem because they don't have as much room, right, for all those internal organs. It's just the long bones don't grow, they don't elongate. But they have a lot of other things that are basically normal sized. It's just their stature is not. So in order to have achondroplasia, you need to have that dominant allele. So if we have an individual who has dwarfism, right, the achondroplasia, that means they're heterozygous, right? But the dominant allele is what gives them achondroplasia. The recessive is for normal height. So if you had someone who's a dwarf and then someone who's normal height mate, half of the offspring are dwarfs and the other half are normal height because it's the recessive allele that's normal in this case. And so there's a 50-50 chance. If a normal height person marries someone with achondroplasia, half their kids will be dwarf and the other half will be normal height. So what if you have two dwarf parents who have kids? Then what? So remember, in order to have dwarfism, you have to be heterozygous. So if we cross two heterozygotes, so these are the eggs and sperm produced, right, by both of these individuals. So this is a monohybrid cross, but we're talking about a lethal dominant characteristic. So if one of the kids is born homozygous dominant, they die. They do not survive. That fetus does not develop. So you're only left with right, the dwarf, and then normal height. So technically, 25% of the offspring die, 50% are dwarf, and 25% are normal height. But in reality, these 25% here, they die. And so that means that 2/3 of the children will be dwarf and 1/3 will be normal height because this condition does not survive to be born. A few more diseases here. Huntington's disease is a degenerative disease of the nervous system. But here's a case where there are no symptoms until you're 35 to 40 years old. So you're phenotypically normal. You do not know you have this condition. So most people, by the time they're 35 or 40 years old, have had kids. So they've passed on this characteristic to their offspring, possibly, right, before they knew they actually had this disease. So sometimes a genetic disorder does not manifest itself for a fairly long time, so you don't know you have it. And unfortunately, you may have had offspring by then and passed on that characteristic to your offspring. So many diseases like heart disease, which I talked about, are multifactorial, a cancer as well. There are certain mutations that make you more prone to certain cancers like breast cancer, but still the environment may have a big impact on whether or not you go on to develop that cancer. And so we really don't know the total effects of genetics and the environment on many conditions. Many conditions may be mostly genetic with a minor effect by the environment, or the environment may have a huge effect on the genetic outcome. There's just a lot of different conditions and a lot of different factors that contribute to them, so it's hard to study. So what to do? Well, we do have tests for many genetic disorders, so it's possible. You can get tested. They do a lot of genetic analyses. You can determine if you have certain mutations which make you at higher risk. I mentioned breast cancer. There's more than one mutation that increases your risk. A common one is the BRCA mutation, the BRCA mutation. And so there are cases where women have a family history of breast cancer, right? Their mother got breast cancer, their sisters got breast cancer, their grandmother, their aunts, they all got breast cancer. And it turns out that they have these mutations. And so there's almost 100% chance they will develop breast cancer. And so they have two options. One is to wait and see, right? Get regular mammograms and breast exams. And if something happens, then you can treat it or have a mastectomy to remove the breast. Some women who know they have basically 100% chance of breast cancer opt to have a double mastectomy, have both breasts removed, before there's any cancer because they know they're going to get it. So there are ways to perform these tests to see what kinds of mutations that you might have. And so you can decide what to do. If modifying your lifestyle, your diet, those kinds of things can help, then you can do that. Or you can choose to do, radical things like have your breasts removed before there's any cancer present. So in a perfect world, we would all know what our genetics are so that we could decide if there's a 25% chance that we'll have a child who's going to be severely affected by a genetic disease, or a 50% chance we can maybe decide not to have kids. So the problem with knowing our genetics is, who do you want to know your genetics? Do you want an insurance company to know your genetics? Because what if they know you're going to develop cancer? You're going to have a heart attack. You're going to develop this disease. Are they going to want to insure you? I mean, there are a lot of ethical problems, with this, because that kind of information can be used against you. And all these people that have done these various genetic tests, you send off a saliva sample or something to get it tested to find out, oh, what's my, origin? Who am I related to? Where did I come from? That company has your genetic profile. What do you think they're going to do with it? Right? So when you send off to get your genetic results, a company, a for-profit company, has your genetic profile. What do you think they're going to do with that? Who gets to see that? Who do you want to see that? Yeah, probably sell it. What if the government has your genetic profile, on file, right? And so they've actually done this where they have a crime and there's DNA evidence, but there's nothing in their files, right? They have no suspects that match, right, that DNA evidence. And they've taken those gene sequences and they've gone to these different companies and they've tried to find relatives who match. And once they find the relatives, then they can narrow down the suspect, because the suspect is related to these people. So even though the suspect has never done any of these family trees or genetic tests, they have relatives who have done them. And so their DNA evidence can be used to match to some of their relatives, and that helps them get caught. So there is a big concern about who gets your genetic profile and what will they do with it. So there are great concerns about that. So any questions or comments? Joa? No, Damilola? All
I
right.
wanted to ask you something, boys. I will stay back.
Okay. All right. So kind of an